2.2. Membrane composition and organization. Lipid bilayer structure. The lipid bilayer forms the fundamental structure of all cellular membranes and acts as a flexible yet stable boundary between the internal environment of the cell and its surroundings. It is composed mainly of phospholipids, which are uniquely suited to membrane formation because each phospholipid is amphipathic, containing both a hydrophilic, water-loving head and hydrophobic, water-fearing, fatty acid tails. The hydrophilic head consists of a glycerol molecule attached to a polar phosphate group, enabling it to interact readily with water molecules in the cytosol and extracellular fluid. In contrast, the hydrophobic tails are long, nonpolar fatty acid chains that avoid water. These tails may be either saturated, creating straight chains that pack tightly and reduce fluidity, or unsaturated, where double bonds cause bends that increase membrane fluidity. When placed in an aqueous environment, phospholipids spontaneously arrange themselves into a bilayer, with the hydrophilic heads facing outward toward the water on both sides of the membrane, and the hydrophobic tails pointing inward, away from water, forming a stable hydrophobic core. This natural arrangement produces a dynamic, double-layered sheet that is essential for maintaining cellular integrity. The bilayer serves as the foundation for the fluid mosaic model, in which phospholipids are free to move laterally, giving the membrane a fluid, flexible character. Embedded within this lipid matrix are proteins, cholesterol molecules, and carbohydrates, which together create a mosaic of components that drift within or on the surface of the bilayer. Insert figure 2.21. The lipid bilayer possesses several important properties that enable it to function effectively as a membrane. One of the most significant is fluidity, which allows the membrane to bend, stretch, and self-repair when damaged. Fluidity is influenced by temperature, by whether fatty acids are saturated or unsaturated, and by the presence of cholesterol in animal cells, which prevents the membrane from becoming too rigid at low temperatures or too fluid at high temperatures. Another key property is selective permeability, which arises from the hydrophobic interior of the bilayer. This interior prevents most polar molecules and ions from crossing freely, while allowing small non-polar molecules such as oxygen and carbon dioxide to diffuse through. Water can pass slowly by osmosis, but larger polar substances, such as glucose and charged ions, rely on membrane proteins for transport. Functionally, the lipid bilayer is essential for maintaining homeostasis, acting as a barrier that regulates what enters and exits the cell. It also enables compartmentalization, allowing organelles such as mitochondria, chloroplasts, and the endoplasmic reticulum to have specialized internal environments suited to their roles. The bilayer further provides a structural platform for membrane proteins, including transport proteins that move substances across the membrane, receptor proteins that detect signals, and enzymes that catalyze reactions at the membrane surface. Its fluid nature also supports dynamic cellular processes such as endocytosis, exocytosis, vesicle formation, and cell movement. Overall, the lipid bilayer is a highly organized yet flexible structure that is crucial for cell survival. Its amphipathic phospholipids create a semi-permeable barrier. Its fluidity allows movement and change, and its interaction with proteins and other molecules supports the diverse functions of the cell membrane. Integral versus peripheral proteins. Cell membranes contain a diverse range of proteins that contribute to transport, communication, recognition, and structural support. These proteins are embedded within or associated with the phospholipid bilayer, but their position and depth of insertion determine whether they are classified as integral or peripheral membrane proteins. Understanding the differences between these two groups is essential for explaining how substances move across membranes and how cells interact with their environment. Integral membrane proteins, also known as intrinsic proteins, are embedded deeply within the lipid bilayer and cannot be removed without disrupting or dissolving the membrane. Most integral proteins span the entire bilayer from one side to the other, and these are specifically called transmembrane proteins. Because the interior of the membrane is hydrophobic, integral proteins contain hydrophobic amino acid regions that anchor them firmly within the phospholipid tails. These hydrophobic regions allow the protein to remain stably positioned, even as the membrane shifts and flows. The parts of the protein extending out into the extracellular fluid, or cytosol, are hydrophilic, enabling interaction with water and dissolved molecules. Integral proteins perform many essential functions in the cell, including acting as channel proteins that form pores for ions and water, carrier proteins that change shape to move substances such as glucose, receptors that bind chemical signals like hormones, and enzymes that catalyze reactions at the membrane's surface. Their structural integration makes them crucial for selective permeability and cell communication. Peripheral membrane proteins, in contrast, are not embedded within the lipid bilayer. Instead, they attach loosely to the membrane surface, usually by interacting with the hydrophilic heads of phospholipids or by binding to the exposed regions of integral proteins. They do not penetrate the hydrophobic core and can be removed relatively easily without damaging the membrane. Peripheral proteins are typically located on the inner surface of the membrane, facing the cytosol, where they play roles in cell signaling, cytoskeleton attachment, and maintaining cell shape. On the outer surface, some peripheral proteins function in cell recognition, often as part of glycoproteins. Many peripheral proteins act as enzymes that accelerate specific reactions near the membrane or assist in anchoring and organizing the cytoskeleton, giving the cell structural support and enabling movement. Insert figure 2.2.2. Overall, the key difference lies in their association with the bilayer. Integral proteins are embedded within the membrane and often span it, while peripheral proteins attach loosely to the membrane surface. Together, these protein types contribute to the dynamic and multifunctional nature of the plasma membrane, supporting transport, communication, recognition, and structural integrity. Carbohydrate chains and their attachment to proteins, lipids. Carbohydrate chains are important components of the cell membrane and play key roles in cell recognition, communication, and protection. These chains, collectively known as the glycocalyx, are found on the external surface of the plasma membrane, where they attach to either proteins or lipids. When carbohydrates bind to membrane proteins, they form glycoproteins, and when they attach to lipids, they form glycolipids. Both structures contribute significantly to the way cells interact with their environment and with one another. Glycoproteins are membrane proteins that have one or more short carbohydrate chains covalently attached to their extracellular regions. These carbohydrate chains are typically branched and composed of monosaccharides, such as glucose, galactose, mannose, or sialic acid. Because the carbohydrate extensions protrude from the cell surface, they act as molecular identification tags. This allows cells to recognize each other, distinguish self from non-self, and coordinate immune responses. For example, blood group antigens, A, B, O, are specific carbohydrate structures found on glycoproteins of red blood cells. In addition to recognition, glycoproteins serve as receptors for hormones, neurotransmitters, and pathogens, helping the cell detect and respond to external signals. The protein portion anchors the molecule in the membrane, while the carbohydrate portion enables precise interactions with other cells and molecules. Glycolipids, on the other hand, are formed when carbohydrate chains attach to phospholipids in the membrane. Like glycoproteins, glycolipids extend outward into the extracellular environment and contribute to cell recognition and communication. Their carbohydrate moieties provide binding sites for specific molecules, help stabilize the membrane, and assist in the formation of tissues by allowing cells of the same type to adhere to one another. Glycolipids are particularly abundant in nerve cells, where they contribute to the electrical insulation and structural organization of neuronal membranes. Together, glycoproteins and glycolipids form the glycocalyx, a carbohydrate-rich layer that coats the exterior of the cell. This layer protects the cell from mechanical damage, assists in cell adhesion, and plays a vital role in the immune system by helping cells identify pathogens and abnormal cells. The arrangement and composition of carbohydrate chains vary between cell types, giving each cell type a unique surface pattern that functions like a biochemical fingerprint. Insert Figure 2.2.3. In summary, carbohydrate chains attached to membrane proteins and lipids serve essential functions in cell recognition, signaling, communication, adhesion, and protection. Their strategic placement on the extracellular surface of the membrane allows them to act as key mediators between the cell and its external environment, making them indispensable to multicellular life.